So far, we have been talking about how proteins are targeted into different organelles. We know that proteins which are inserted in the ER membrane or which are inserted in the lumen of ER, they actually are transferred to the Golgi apparatus. That transfer is not direct. The protein doesn't come out in the cytosol exactly. Rather than that, it is enclosed in a vesicle, and that vesicle buds off from the endoplasmic reticulum and fuses with the Golgi apparatus. So these proteins are carried in a container, if you will, and that container is taken from ER to Golgi. It happens in reverse also. So now we are going to focus on protein targeting via vesicles. Let me refresh your memory by talking about Golgi apparatus first. The Golgi complex is a major site of carbohydrate synthesis as well as sorting and dispatching station for products of endoplasmic reticulum, which it receives from the endoplasmic reticulum. Many of cells' polysaccharides are made in the Golgi apparatus, including pectin, hemicellulose of the cell wall in plants, and most of the glycosamine glycans of the extracellular matrix in animals. Golgi apparatus also lies on the exit route from ER, and a large portion of carbohydrates that it makes are attached as oligosaccharide side chains to many proteins and lipids that ER is sending to the Golgi apparatus. Addition of carbohydrate molecules to proteins serves a very important function. For example, the proteins that are destined to lysosomes, they have specific arrangement of these carbohydrates on their surface. That also is facilitated, some part of it also occurs in Golgi apparatus. Most proteins and lipids, after acquiring their appropriate oligosaccharides in the Golgi, are recognized in other ways for targeting into the transport vesicles going to other destinations. As I mentioned, one of the destinations for proteins which are being changed or being altered in Golgi apparatus, one of their destinations is also lysosome. Proteins that enter the ER and are destined for Golgi apparatus are packaged into transport vesicles which bud off from the specialized regions of ER called ER exit sites, whose membranes lack bound ribosomes. So these membranes of ER, they're specialized. Basically, their purpose is to form these vesicles, and these vesicles, once they're formed, they fuse with the Golgi apparatus. And these sites, these special exit sites, they lack ribosomes, which we have talked about, the smooth ER and the rough endoplasmic reticulum. Some cargo proteins are actively recruited into such vesicles where they become concentrated. Of course, if ER is sending proteins to Golgi apparatus, it needs to make sure that only proteins which are supposed to function in Golgi apparatus are packaged into these vesicles. There are proteins, subset of proteins which ER requires for its functioning. Those proteins should not go to the Golgi apparatus. The proteins which have to go to Golgi apparatus, they have exit or transport signals on their surface which are recognized by complementary receptor proteins. These receptor proteins are special. They are embedded in the endoplasmic reticulum. So in the lumen of endoplasmic reticulum, these proteins which have exit signals binds these receptors, and then these receptors, they have the ability to come together and form basically a small vesicle with the help of special proteins which assist in the formation of this vesicle. In this case, those proteins are referred to as COP2 proteins. So we are going to talk about these proteins in more detail subsequently, but here I wanted to just mention how the vesicle formation is occurring. It is doing two things. It is concentrating proteins that are going to be transferred to the next compartment of the cell, and secondly, it is also, these proteins play another function. They recruit other special proteins, coat proteins, that facilitate the formation of the vesicle. Cargo proteins displaying the transport signal on their surface are recognized by complementary receptor proteins which trap these proteins, and these proteins are then taken to the Golgi apparatus. Here, for example, this is a nice diagram to show you how the system works. This is the lumen of the endoplasmic reticulum, and you can see there are many proteins. Some of these proteins which have the signal for exit signal, they will bind the special receptor proteins. For example, here is a soluble cargo protein that needs to go to Golgi apparatus. It will bind a receptor. The binding domain of this receptor is actually inside the lumen of endoplasmic reticulum, and there is a cytosolic domain of this receptor. Once this interaction happens, there's a conformational change in the cytoplasmic domain of this receptor, and which is recognized by additional proteins. And these proteins basically, first of all, they concentrate all these receptors which have active cytoplasmic domain. They concentrate them in one location, and also additional proteins. For example, here we were talking about COP2 proteins. These are special proteins. They assemble at the cytoplasmic domain of these receptors and forcing the formation of a vesicle. The vesicles of animal cells leaving Golgi destined for plasma membrane are rich in cholesterol. So here's an additional mechanism which allows concentration of specific proteins in a specific region of a membrane. The lipid bilayer of cholesterol-derived vesicles is thicker than that of Golgi membrane itself. So after Golgi has received packages, different proteins from endoplasmic reticulum, now it needs to send some of them to the surface of the cell. For example, we have talked about T cell receptors or B cell receptors. These proteins, they have larger or slightly longer transmembrane domains which can insert specifically, preferentially into the certain regions of Golgi apparatus which are rich in cholesterol. Because these regions are rich in cholesterol, these regions are thicker than the rest of the Golgi membrane itself. Transmembrane proteins with longer segments, which are long enough to span this thicker portion of the Golgi membrane, will preferentially be entrapped in those regions. Generally, ER resident transmembrane proteins have short transmembrane segments around 15 amino acids. However, the proteins which have around 20 to 25 amino acids are specifically preferentially embedded in the regions of Golgi membrane which is rich in cholesterol. For example, you can see this part of the Golgi membrane, it does not have cholesterol, so this region is thinner. And transmembrane proteins with shorter transmembrane segment or alpha helices can be inserted in this region. However, proteins with longer transmembrane domains are preferentially embedded in the region of Golgi apparatus which is rich in cholesterol. So this basically region which is rich in cholesterol and has proteins embedded in it is also called the lipid raft. So there's an additional mechanism for entrapping proteins which are destined for example, in the plasma membrane. Coming back to exit from ER, these proteins first of all have to be folded properly in order for them to be transported to the Golgi apparatus. Misfolded or incomplete assembled proteins are retained in ER where they are either degraded or other proteins help them fold properly. For example, there is a protein called ERGIC53. This protein serves as a receptor for packaging some secretory proteins into COP2 coated vesicles. The role of this protein was identified because people who have a mutation, which is heritable mutation, have lower levels of two secreted blood clotting factors, factor 5 and factor 8. Therefore, they bleed excessively. So I just wanted to emphasize that it is very important, crucial for the viability of cell to select specific proteins going to specific organelles. This helps cell maintain its normal function.